So I'm just going to do a little introductions for each of our speakers. As Robert mentioned, he is the director here at the MSSR and the CNSI. He obtained his PhD while working on directed molecular evolution of catalytic antibodies at the University of Lausanne in Switzerland. Joined Novartis Institute in La Jolla in 2001, where he worked on the development of next-generation screening methodologies. He has resided at UCLA since 2004. He oversees all drug discovery as well as functional genomics projects at the MSSR and feels at home at the interface of chemistry, biology, and engineering. His diverse research interests include drug discovery, development, nanotoxicity, and high-throughput screening, and they are reflected in his many publications, book chapters, and patents. Thank you, Robert.
Here's an overview of my talk today. I'll start with an introduction to the MSSR, to the different resources we have here for you that pertain to drug discovery itself as well as functional genomics. Then I'll give a cursory overview of toxicology for drug development. And then I'll show you as well how you can use the resources at the MSSR for toxicity testing. And I'll give you two examples. The first one deals with genotoxicity, and the second one is an in vivo screen using zebrafish and ROS screening. We do two things at the MSSR. The first one is high-throughput screening. That means that we provide consultation, assay development, and validation services. We can execute your HTS campaign as well, and we provide result verification. We have an open access policy. This means that everyone is equally welcome. And secondly, we perform as well our fair share of tech development. That means that we identify bottlenecks in HTS, devise, build, and implement novel solutions to these bottlenecks. I'll come back to that later on. Tiny bit about the origins of high-throughput screening. I brought you two pictures here, one of a laboratory around 1900 and one around 2000. Maybe it's more 1990, 1980s. Looks a little dated. But the short of that is when you really look at the number of people in these two pictures and the floor plan, it looks actually all very, very similar. So the short of this is, the gist really is that the Industrial Revolution has not been realized in most laboratories. Things are still being done by hand, and results come in relatively slowly if you compare that, for example, with microprocessors. Now, around 1980, that picture's changed very, very drastically. Big Pharma looked for a more potent, more intelligent way to discover novel medicines. And that, for them, was high-throughput screening. High-throughput screening can be loosely defined as an automated repetitive execution of an assay for more than 10,000 times per diem. So the short of that is, what can you do really with HTS or high-throughput screening? Well, we talked already about drug discovery, where you might be screening small molecule libraries that could be targeted libraries against GPCRs, proteases, kinases, and so on. And there are drug-like libraries and very diverse libraries. Well, high-throughput screening lends itself as well as a very, very versatile research tool. You could, for example, use chemical genomics, where instead of traditional mutagenesis, you use annotated drugs to shut down key pathways or key enzymes and study the phenotype that this chemical knockout induces. High-throughput screening is as well very useful for functional genomics. Here you use genome editing technology such as knockout with Si or lentiviral SH constructs or CRISPR in order to edit the genome and knock out the function of certain genes. Again, you study the phenotype and see how it pertains to, for example, a given disease. It's very useful to have as well cDNA overexpression screening, and that would enable you to perform gain-of-function screening. With that being said, high-throughput screening made as well forays into materials discovery, such as high-throughput chemistry, high-throughput quality control, And last but not least, and most relevant for today's topic, materials characterization using automated toxicity profiling and nanotoxicology. Here at the MSSR, we have a heavy focus on high-content screening. High-content screening is basically fully automated microscopy coupled with image recognition software. And what does this look like? Here is an image of a 384 well plate, and a couple of wells are zoomed out. And what you see here, these little colonies of cells that are highlighted in green, those are human embryonic stem cells. We were looking for compounds that enable these stem cells to survive better. So for example, this well here has a number of these green colonies and would be scored by the software as a hit, whereas this well here has very few. What's as well interesting to note is you see maybe in blue here the nuclei of all of the other cells in the background, is that you can get a cell population that are by far not the majority of the cells using high-content screening. You can screen as well for rare cellular events, you can screen for morphology changes. All of that is available to you using high-content screening. So with that being said, the MSSR started out about 10 years ago, and we started with small molecules. That was really, and still is, our bread and butter. Very rapidly, we added in functional genomics and materials discovery and nanotoxicology. Nanotoxicology we perform in conjunction with and for the UCCIN, the Center for Environmental Implications of Nanotechnology here in the house, in the CNSI. Small molecule is something that many different departments here at UCLA are interested in. We have even customers from as far away as England and India. But the CTSI is one of our biggest partners here in the small molecule arena. And last but not least, we're here in the CNSI, so this is where molecules, nanomaterials are being made and tested. What do we provide at the MSSR? We provide three things. Number one, the equipment. That means robotics, plate readers, high-content readers, an entire infrastructure that you might need. Secondly, we provide the libraries, such as small molecules, functional genomics libraries, as you see it here, and then as well the know-how. And how do you work with the MSSR? Well, typically, the PI comes in with a biological question or indication, and you may or may not have already an assay, or at least you have an idea for an assay. And we can take that and adapt your assay to high throughput. This means that we perform technical validation, miniaturization of your assay, typically at least to 384 well, and then statistical validation as well, and trial run on a small library, typically for pharmacological validation. Then we execute the assay, and we perform statistical analysis of the assay data. And the results then go back to the PI. So with that, the role of the MSSR is completed. A little bit about our libraries. We have about 200,000 small molecules here available for you. And they range from small libraries such as chemical genomics probes over FDA-approved drugs for repurposing to natural products, in-house collections, and the libraries keep getting larger and larger and larger. So you come in for targeted libraries, lead-like, diverse, and smart libraries. We have as well 200,000 siRNAs, cDNAs, and shRNAs to screen from. We have an siRNA for the mouse and human genome. We have a cDNA overexpression library that's 16,000 clones strong. And we have as well a genome-wide shRNA library. And CRISPR sets are right now being implemented and should be available for screening in the next 4 to 6 weeks. Something that maybe sets us apart from most other facilities is our ability to take individual clones from our larger sets and make them into targeted sub-libraries. So let's say, for example, you have a specific target you need to validate, or you have a certain pathway that you're interested in. We can take all of the pertaining clones out of our libraries, automatically erase them out on a qbot, and then make you ready to use DNA sets that you can either then use for reverse transfection and screening, or we can as well prepare those in lentiviral formats for transduction should your cells require that. In terms of readouts, we have pretty much any label for you available, be this now luminescence, alpha fluorescence, or time-resolved modalities. We can perform as well kinetic fluorescent readouts using Flipper or FlexStation. And I mentioned already our high content screening capabilities. Maybe I want to add that we have phase contrast and epifluorescence, as well as laser scanning cytometry available. We are approved all the way up to BSL-2+, so you can bring us pretty much any pathogen as long as it's not BSL-3. So now I want to shift gears and talk a little bit about the cursory overview of toxicology for drug development. I think most of you are very familiar with the drug discovery process, where you start out with target discovery, you're trying to find the disease-modifying target, try to validate this target, and then you're trying to find a small molecule that does the trick and hopefully cures the disease. Afterwards, you're coming in with medicinal chemistry to improve the molecule, and then from there it's PK, ADME, tox, and then eventually you're going into pre-IND and IND-enabling studies in phase 1, 2, 3. Now, something that's important here is when you have your discovery part and you're shifting over into the preclinical phase 1, 2, 3 part, you're shifting from discovery toxicology into regulatory toxicology. And that means, on the other hand, that you now have to deal with GLP requirements, at least if you want to use the results for IND-enabling studies later on. So that becomes a whole another ballgame. But what you're really trying to do here in the discovery part is you're trying to take down the hits that you get from your screenings that are in the hundreds or even thousands down to 1, 2, 3 hits, basically typically Series A and Series B compound, that then eventually can be translated into human. And central to that question here that you have to answer is my small molecule candidate toxic? Now, Paracelsus around 1500 knew already, die Menge macht das Gift, or in plain old English, the dose makes the poison. So in other words, the man had a very keen appreciation for therapeutic index. Therapeutic index is basically the ratio between the concentration at which you see a desired effect versus an undesired effect. Normally, you want this difference to be as big as possible. Typically, that has been measured in vivo with LD50s, but the same concept applies to in vitro measurements, and that's what we're dealing with here today in our symposium. To make things a little bit worse, there's many, many different toxicity types. Basically, as many organs as you have in a human, you have different toxicities. It could be neurotoxicity, hepatotoxicity, or genotoxicity, and these are not mutually exclusive. Now, what you're trying with in vitro assays is you're trying to take an in vitro assay, maybe on a cell panel for cytotoxicity, and you're trying to cover the bases. Neuronal cell assay, for example, for neurotoxicity, immune for immunotoxicity, and so on and so forth. Or, for example, DNA damage assays for genotoxicity. So that's the general idea here. And I think today we mostly will talk about actually as well cytotoxicity, hepatocytotoxicity, DNA damage, and oxidative stress. There's many problems with the current picture of in vitro toxicology. Typically, they stem from the fact that small molecules have very unique properties. In other words, they have many potential modes of toxicity that are possible, and it's not clear a priori which of these routes the molecule will take and how it's going to affect an organism. Secondly, there's many different labs that use different toxicity assays, and that makes it very difficult to compare the data. And last but not least, there is a general lack of the standardization of these assays. So I hope in our Q&A session we'll be able to have a discussion about what would be the best assays to run, and how can we standardize the assay process itself a little bit. tiny bit about the in vitro toxicity resources at the MSSR. So I brought you here a very generic panel of assays that you might want to run. Typically, there is an Ames or micronuclei test for genotoxicity, HERC for cardiotoxicity, P450 assays for drug-drug interaction, hepatotoxicity, general cytotoxicity, and then maybe some others depending on your project. Then, of course, you might want to run a promiscuity assay. This means you want to make sure that your compound doesn't bind to any old receptor. That means that you will be screening a large panel of assays. That's normally outsourced. In terms of what you can do of these assays at the MSSR, it's really up to you. There's really no limitations because we can help you any step of the way. Typically, you'll start out with a selection of your compounds for follow-up from the hits. And you can do this in an automated fashion using our CDD platform. Then you'll be making your dilution plates from compound powder. And we have robotics and the MSSR staff who can show you how this works precisely. And then the assembly and execution of the assay, and we can relatively easily roll out 20,000 wells in a day in dose response. Last but not least, there's automated IC50 calculations, and CDD does this very nicely. So with that being said, I want to show you two examples of what you can do in terms of toxicity assays at the MSSR. The first one is a genotoxicity assay using laser scanning cytometry. That's an EPA collaboration. Genotoxicity takes a special place amongst all toxicities, particularly because the actual carcinogenicity testing happens after phase 2. It's very expensive, and the problem really is that since rodents do get spontaneous cancers, it's very, very hard to execute and to interpret the data. So part of the project, part of the explanation I should maybe give you and how the project came to me was someone came to me, Dr. Hirschman came to me and said, "Are you aware of a thyroid cancer epidemic?" And I said, "No, I've never heard of that." And then he said, "Well, tell you what, around the 70s to the 80s, thyroid cancer incidence was pretty flatlined. And then around the 90s, it started to tick up, and no one knows why. People thought, well, maybe this is because we are better able to detect these now. But then around 2000, you see an almost exponential rise of the incidence here, and people start to get really, really worried. So when you think about that, and you have to think as well about the fact that thyroid cancer takes 25 years to actually develop, it's not very easy to make a causative conclusion here. What is it that causes this? Part of the equation is that our assays are really less than perfect. We have, for example, the Ames test that you could run in high throughput. And that's a bacterial recombination test that's very dated. Then there's a Vitro Tox test that runs on a REC-N promoter. And then the green screen, that's mammalian, uses the GET45 reporter, and that's basically a DNA damage assay. What makes the whole thing a little problematic is that we have 40% of the genotoxic compounds slipping by the Ames test, and about 80% get detected with a green screen. So that's not ideal. I think the real problem here is, especially with the Ames test, that we're trying to detect one productive mutation in a 4.6 or more gigabase genome. So that's not very productive. But what would be a better way of doing that would be to detect really the primary damage event. So if you have a look at the cartoon here of what happens after you apply a genotoxic agent to the DNA that you see here wound around the histone, specifically around histone H2X, you will induce a double-strand break, and then you have the phosphorylation of serine 139, also called gamma. And this serine, this phosphorylated serine, is actually the adapter protein that recruits all of the other repair proteins. And you can quantify that very nicely. Typically, what you do, in our case, you would use thyroid cells, plate those out, and come in with a compound, and then come in with a primary antibody against phosphoserine. We would wash that, come in with a secondary antibody, and that's something we can detect with laser scanning cytometry. And that's not a reporter assay. We're really quantifying a biological response here, which is much more closer to what we really need this to be. So this is what this looks like. Here on top, we have propylium iodide. Here on the left-hand side, we have the solvent only. Here we have etoposide, a positive control. Etoposide is a DNA gyrase inhibitor. And as you see here in the gamma H2AX on the left-hand side, you see a little bit of background. And here you see the cells really lighting up. This is what this looks like when you quantify it. Here's just the total number of cells. And here you see a really nice shift in the gamma H2AX signal, shift over to the right-hand side. With a Z prime of 0.8, you can measure this very nicely. When you have a look at the fraction of gamma H2AX positive cells, you get a really nice dose response. You have to normalize the total number of cells because otherwise the error bars become very big because genotoxicity affects cell division and so on and so forth. But once you've normalized, it's very beautiful. The general workflow is relatively easy. You start out with your compound, you make your dose response plates, apply, and run your assay. And what we found was that, I can't give you the name of the pesticide right now, we're writing up the paper, that a lot of compounds, especially pesticides, are very good at inducing DNA double-strand breaks. So this is the picture after 2 hours. After 8 hours, the response is very nicely established. And after 24 hours, it looks like this. It surprised me a little bit that the response is so sustained. And let me tell you, it looks identical after 48 and 72 hours, which prompted me to make sure that I'm going to be eating organic from now on. Okay, let me shift gears here and talk a little bit about zebrafish screening. Zebrafish screening is one of the most powerful in vivo systems that is amenable to high throughput. Zebrafish, basically, when you take the fertilized eggs from the zebrafish, you can put them into 96-bar plates, and you can then expose them to either nanoparticles or small molecules or whatever else you need to do. And then you can have different modes of readout. Here at the MSSR, we have an image express with phase contrast. We have as well an Acumen, laser scanning cytometer. And that's a very nice, very easy workflow. So the only thing that's a little bit of a pain is actually putting the eggs into the plate, but I'll come back to that in a moment. What makes zebrafish so nice is that you have many targets that are conserved across species, and they're transparent, so the imaging works really, really well. You can construct transgenic fish with relative ease, and last but not least, you have all of the developmental targets accessible. Zebrafish develop fully within days, so you can get at all of the targets that normally, because you're testing only adult mice, that are normally inaccessible to you. It's very, very versatile. Here we have dopaminergic neurons that are stained with GFP. Here we have some immune cells. This is angiogenesis marker. And this here is smooth muscle. So you can light up any part of an organ or any cell types that you'd like to and hone in on it. Here's a very simple screen. That's a simple hatching phenotype. Here's a control. And here is increasing amounts of copper oxide nanoparticles. And you see here 0.1 ppm, they still hatch. And then as you go higher, they don't anymore. That's a very simple, very powerful approach. For these nanoparticles, you're dealing with a lot of background. You need to do a lot of image processing, but that can be dealt with. The same can apply as well to small molecules. Shifting over, I talked as well about constructs you can make in zebrafish. So for example, here we have a heat shock protein 70 EGFP fusion. And here's what this looks like. We've never really found out about the eyes. The eyes of the zebrafish are always lighting up. I think they're reflective or something. But if you, for example, expose the fish to heat, it lights up very nicely. And then if you, for example, use cadmium chloride or copper, zinc, you see the fish light up very nicely. And these are metals that are known to induce oxidative stress. Nickel and cobalt less so, as was to be expected. But what makes the system so powerful is actually that the total amount of fluorescence that you're getting out of the fish corresponds very nicely with the mRNA levels. So you basically can get out of your images the mRNA level surrogates, which is a very powerful way of doing this. So I talked a little bit about having to array out zebrafish embryos, and it's very, very painful. It's very, very slow. So CG from the CEAN and I built this little construction here, and that's a robotic arm with a camera, and here's the zebrafish embryos and a little petri dish. And what this does is it takes an image, calculates the coordinates of a zebrafish egg, sucks them up into the pipette, and erases them out. And that's what this looks like. I hope this video is going to play. Maybe not. But basically, we'll just have to then imagine it does. Basically, what it does is it erases very quickly all of the embryos in the petri dish into all of the 96 or 384 well plates here, and gets rid of this manual step so that we can perform high throughput screening in zebrafish. So with that, I want to conclude and get out my acknowledgments, especially to the JCCC, the CNSI, the UCLA Shared Resource Consortium, and to the MSSR. I want to acknowledge Dr. Bradley, Brian, and Bobby, the technicians, Dr. Hirschman for the H2AX project, zebrafish, Dr. Nell, and Dr. Lin. And thank you for your attention. I'll take questions. Pardon me? Again?
Can you provide some examples of how faculty have funded projects through the MSSR?
Can you be more specific what you mean by funded?
All the projects coming from grants that propose high throughput screening, or some of the other mechanisms that faculty have used to pay for these studies.
Right. So there's actually, there's actually a lot of projects that we have collaborated with faculty. I think someone summarized that all of the grants that we've helped brought in, and that was around 120 million total. So we normally are part of their grants, we help them write the grants, we help them bring in all of the different technologies as they need it, and so on and so forth.
So thinking about how to design experiments for the core, one might approach you about the design of a high throughput screen even before the grant goes in?
Yeah, absolutely. So you can come to me anytime and ask your questions with anything you need. Very frequently as well, people come to us and say, I don't want to do anything high throughput, I just need, say, this plate measured, and I need the number of cells counted. And if it's just a one-off, even that's doable, very easy. Yes?
So in the high throughput screening of the zebrafish,
Mm-hmm.
can you just we don't do any fish. Do you grow this fish? How do you do this experiment?
Where do we get our fish from? Yes. There's actually 2 facilities on campus that grow fish, and that are as well recharge facilities. And that's where we get our fish from.
So how do you do this in a high throughput sequence, high throughput fashion?
So the fish normally are very prolific. They actually lay hundreds of eggs so that out of a few fish, you get many thousand eggs. Per day. Anything else? Okay. All right, then we should probably ship.
One more question. So, is there some way of synchronizing the development so you have the eggs progressing to embryos, and how do you pick the right ones and start at the same place?
Right. That's actually an interesting question. So what I've been told, because I don't normally handle the zebrafish egg production, is that they actually pretty much all synchronize according to light, to the circadian rhythm. So in as much as that, it occurs naturally, always roughly at the same time. You just have to be quick enough to erase them out so that they don't get old.